Hello everyone and welcome to my channel ATGC of Genetics. This is Trisha and today we'll be discussing the ATGC of Sickle Cell Disease. So let's start off with looking at what all we'll be discussing in this video. So we'll be starting off with an introduction to Sickle Cell Disease. Then we'll be finding out what's the cause behind SCD and understanding its pathology. Then we'll go on to understand its inheritance, symptoms or complications, diagnosis, treatment, and the different types of sickle cell disease. So let's get started. So sickle cell disease is the most inherited and the most common blood disorder. It is genetic in nature, which means that it is not something which you can catch later in life, but it is something which is in your genes. It is caused due to abnormal hemoglobin produced in your body, and the hemoglobin is sickle shaped in this case. There are various different variations of diseases which come under the umbrella of sickle cell disease, which we'll be discussing towards the end of the video. Let's understand what do we mean by the sickling of hemoglobin. So in normal cases where a person does not have sickle cell disease, you would see that the hemoglobin, which is the molecule present in RBC of a body responsible for providing oxygen to each and every part of a body, is round, smooth, and flexible. Whereas in the case of sickle cell disease, you would find that the hemoglobin is sickle shaped, and thus it tends to become rigid and sticky, and it tends to stick together. This in turn causes blocking of blood vessels inside the body. The sickling of cells is not present throughout the lifespan of the person, but it is triggered by certain factors. These could be low oxygen levels, increased acidity, and low volume of blood or dehydration. So here on screen you can observe the prevalence of sickle cell disease throughout the globe. You would observe that a lot of countries in Africa, then there's India, some part of South America. These areas have a higher rate of sickle cell anemia in their region. It's also important to note that these are countries which are endemic to malaria. So could these two facts be relatable? Indeed they are. This is because the sickle shape of our RBC provides resistance to our body from catching malaria. So since malaria is endemic in these regions, our body has evolved to form the sickle cell hemoglobin, which provides us resistance to malaria, but it definitely causes a lot of complications as well. So let's now understand the pathology of sickle cell disease. For this, we first need to understand what is DNA. DNA, which is deoxyribonucleic acid, is the genetic material inside a body. And you can imagine this with the help of a necklace of pearls. If you would have seen my previous video, you can skip through this part, but if you are new here, then I would recommend you watching this. So if you imagine a pearl necklace, you would observe that there's a thread and there are pearls pierced in, right? So you can imagine the thread as being the backbone of our DNA and the pearls as the basis on our DNA. And the bases can be adenine, guanine, thymine, or cytosine, which is AGTC. So a simple representation of a DNA could be like this. They're just arranged in a specific order, and it's important to note that their order, for example in the screen A, C, G, T, G, G, C, A, C, this particular order is very important for all the proteins synthesized in our body, as DNA is what gives rise to protein later on in our body. So if this particular sequence differs even by a letter, or even if there's some repetition in the sequence, some deletion, the proteins in our body in most of the cases are affected by it, and we observe changes or diseases. A gene is something which is responsible for producing a certain protein inside our body. So the gene could be responsible for producing a protein such as insulin, maybe for blood sugar level control, or there are various different proteins produced for every function inside the body. So a gene could be anywhere in the DNA. It could be, for example, here or here. So let's just assume that the second circle is the hemoglobin beta gene, which plays a vital role in predicting where the person would have sickle cell disease or not. So let's zoom into the hemoglobin beta gene. So in this image, you can see two different scenarios. The first, in which the person does not have sickle cell disease, and the second, where the person has sickle cell disease. So in the first case, you see that there's GAG in order, and this GAG gives rise to a particular amino acid glutamine. Whereas in the second case, just the replacement of A with T, which gives rise to GTC, produces a completely different amino acid, which is valine, which changes the configuration of the protein produced completely. And this different protein which is produced is the reason why the hemoglobin inside the RBC of our bodies tend to sickle. So let's now understand the inheritance of the hemoglobin beta gene, which is the gene responsible for sickle cell disease in a body. So here we observe that the gene is inherited in autosomal recessive fashion. So before understanding what is autosomal recessive, it's important to understand two different terms. The first being sickle cell trait and the second being sickle cell disease. So here in this picture, you can see that there's a father and a mother and they have 4 different children. So you can see that the father and the mother have one chromosome which has the abnormal hemoglobin beta gene and the second which has a normal hemoglobin beta gene. I think you would know that all the chromosomes in our body exist in pairs. So for chromosome number 11, on which the hemoglobin beta gene resides, it again, as it exists in pair, it has 2 different alleles for the same gene. So in the case of sickle cell trait, the person just has one allele or only one chromosome which has abnormal hemoglobin beta gene, which is responsible for producing sickle cell disease. And the other allele or on the other chromosome, there resides a normal hemoglobin beta gene, which does not produce SCD. Whereas in case of sickle cell disease, the person has both the alleles which are abnormal or which give rise to a protein which causes sickle cell disease. So what's the difference between the two? What happens if you just have only one chromosome or one allele which is affected? So in sickle cell trait, the person does not have the symptoms of SCD, but he or she is just a carrier of the disease. A carrier is basically someone who can pass on that particular gene to their children. Alright, so in this picture you can see that these two people have 4 children. In the first children, the child inherits one chromosome which is normal and another chromosome from the mother which is also normal. Thus, the child does not have sickle cell disease. In the second child, the child inherits a normal chromosome from his father and an abnormal chromosome from the mother. So here you would see that the child has sickle cell trait. In the third case, again you would see that the child inherits a normal and an abnormal chromosome from their parents, and Here you would see that again the child has sickle cell trait. And in the fourth child, the child unfortunately inherits both the defected alleles or defected chromosomes from his or her parents. So here the child would have sickle cell disease. Here in particular, sickle cell anemia. So here in this image, again you can see different cases of inheritance of hemoglobin beta gene. As there are two conditions here, which is sickle cell trait and sickle cell disease, the inheritance pattern varies and is a bit complicated. So you can see that if one parent has the trait, if the other parent is normal, then there's 50% of the chances of having sickle cell disease for the children. Then if in the second case you can see that the father has sickle cell disease and the mother is normal. So here in all the kids, definitely you would find that these children would be sickle cell carriers, or they would have the sickle cell trait. In the third case, you can see that both the parents have sickle cell trait. So here 50% of the children would have sickle cell trait, 25% would be affected by sickle cell disease, and 25 could be normal. And in the fourth case, if one of the parents has sickle cell disease and the other parent has sickle cell trait, 50% of the children would have sickle cell trait and 50% would have sickle cell disease. And if both the parents have sickle cell disease, as you can see in the middle, then definitely all the children will have sickle cell disease. It's also important to note that it is called autosomal recessive because if only one chromosome or only one abnormal gene is present, the disease does not show the symptom, or the disease is not present basically. It is only present when both the alleles on both the chromosomes are abnormal. So let us now discuss the symptoms or the complications observed in sickle cell disease. So the most common complication or the symptoms seen in SCD is anemia. You might recall from what we discussed earlier in this video that the sickle cell RBCs tend to become very rigid and sticky, and thus they block blood vessels. So if they block the blood vessels in the spleen, which is the organ responsible for replacing our RBCs, the RBCs thus cannot be replaced. If you might not know, the RBCs have a lifespan of about 120 days only. So after 120 days, they need to be recycled by the spleen so that they can be produced more number newly, and thus they can supply blood to every organ of your body. So if in case their replacement is halted or the function of the spleen is disrupted, anemia is observed. The second symptom is hand or foot syndrome. This is one of the most common and the first symptoms observed in babies who have sickle cell disease. Here, the blood vessels of the hand or feet can be blocked, and thus this causes swelling or enlargement of the hand or feet. So, this is thus characterized by the name hand or foot syndrome. The third symptom can be splenic sequestration. So this again is caused by the blockage of blood vessels in the spleen, and here in this condition the pain becomes so severe that the organ needs to be removed by operation. The fourth symptom could be infections. As we discussed in the previous point that the spleen needs to be removed, removal of the spleen could result in our body to become vulnerable to a lot of infections. And that's the fourth symptom. The next symptom could be delayed growth. It is observed that the children who have sickle cell disease tend to exhibit delayed growth, although they attain almost the same height as other individuals who do not have sickle cell disease by the time they become adults. The next symptom is eye problems. So, as we discussed earlier, that the sticky sickle shaped RBCs tend to block the blood vessels. So when they tend to block the blood vessels of the eye. We have eye problems. When they tend to block the blood vessels of the brain, we see neurological complications. If they block blood vessels of the heart, we could observe heart disease. If they block the lung, then it could give rise to lung disease. And in case of the sticky RBCs blocking the penis, we could observe a condition called as priapism. Here it causes unwanted erection, which could be very painful. The next symptom is gallstones. This is not something which is caused by blocking of blood vessels, but this arises due to the fact that when the RBC is broken down, it releases a byproduct called bilirubin, which is responsible for forming gallstones inside the body. And the next symptom could be sickle chest syndrome, which is characterized by a lot of pain observed in the chest. Let's now discuss the diagnosis of sickle cell disease. So there exists different kind of tests through which we can make out if the RBC is present inside the person and sickle shape or not. These tests could be hemoglobin electrophoresis, sickle solubility, etc. But in most of the cases, especially in countries like the US, the condition won't arise when you have to yourself go to a doctor and get yourself checked for SCD. This is because in those countries we have something called newborn screening. So this is when the newborn baby is tested for a specific for a certain number of genetic diseases, and in the US, for example, sickle cell disease is one of them. So as soon as the baby is born, he would be tested for it and soon would be diagnosed. Let us now discuss the treatment for sickle cell disease. So one of them could be rehydration with intravenous fluids. As we discussed earlier, that one of the factors causing sickling of the cells is due to dehydration. So this is one of the measures which could be taken to reduce the symptoms. The second being blood transfusions with healthy RBCs. The third being treatment of infections, because the stress of infections could lead to further sickle cell crisis. So the treatment of infections could be a very good measure to cope with sickle cell disease. The fourth being supplemental oxygen, which helps the person to breathe. Next being pain medication, drugs to increase fetal hemoglobin, and last being bone marrow transplantation, where the bone marrow is transplanted as it would then help produce RBCs which are in normal state. Also, this is done perfectly for children, like it is better for children below the age of 16 to find the right match with the donor. At the end, let's now discuss the different types of sickle cell disease. So the first being sickle cell anemia or hemoglobin SS disease. This is the most common type of sickle cell disease, and it occurs when you inherit a copy of the hemoglobin S gene from both the parents. It has the worst symptoms of all types and has a very high rate of symptoms. The second being hemoglobin SC disease. This is the second most common type of sickle cell disease. It occurs when you inherit one hemoglobin C gene and one hemoglobin S gene from the other parent. The symptoms are similar to hemoglobin SS disease, however the anemia is less severe. The third type being beta thalassemia minor. Here this condition affects the beta hemoglobin gene production. So the hemoglobin found inside the body is basically made of 4 subunits, 2 of which are alpha and 2 are beta. So here the beta globin gene production is affected. So the size of the RBC is reduced because less beta protein is made. And here another gene called beta thalassemia gene has a role to play apart from HBS gene. So this condition arises if you inherit a beta thalassemia gene from one parent and a hemoglobin S gene from another parent. the symptoms are not as severe. The fourth type being beta thalassemia major. Here again, the beta thalassemia gene or the beta globin gene is involved along with the HbS gene. It has similar symptoms to HbS anemia, and the symptoms here sometimes could be more severe, and it is also associated with the poorer prognosis. The next type being hemoglobin SO condition, and the last being hemoglobin SE condition. The last two conditions are very rare and usually they do not have severe symptoms. So that's all for today's video. I hope this video was informative. If you have any queries, any doubts, if you'd like for me to go over another topic in detail, then please do let me know in the comment section below or on Instagram, or you can mail me as well. Thank you so much.